Good afternoon and good evening, everyone. Welcome to today's webinar. My name is Stella Dlamini, and I'm a public health program manager at the Hepatitis B Foundation. And I also manage our Hepatitis Delta Connect program, which is hosting today's session. So for today's webinar, we're very excited to be sharing highlights from the Delta Cure and AASLD 2025 meetings, where we will be discussing some of the progress that we've seen in hepatitis delta diagnostics, as well as their therapeutic pipeline for delta. And before we get started in our conversation today, just a few housekeeping items. So all members of the audience today will be on mute, but please feel free to use the chat to let us know who you are, say hi, and if you have any technical difficulties, please let us know, and we'll try and get that fixed as soon as possible. While we hear from our speakers today, if you have any questions pop up, please leave them in the Q&A box, and our speakers will attend to the questions at the end of our discussion. I am very excited to introduce our panelists for today. So we will be hearing from Dr. Gish and Dr. Agarwal. So Dr. Gish is currently the medical director of the Hepatitis B Foundation. He's also a clinical professor of medicine at Loma Linda University and an adjunct professor of medicine at the University of Nevada School of Medicine. And we'll also hear from Dr. Kosh Agarwal, who is a consultant hepatologist and a transplant physician at the Institute of Liver Studies at King's College in London. So thank you both for joining us today. And I also would like to introduce Super Shanskol, who is a founding member of Hep B Companion, which is a nonprofit that advocates for people that live with hepatitis B. And Super will be helping moderate today's session as well. And this is a brief run through of the agenda for today. So we will be discussing hepatitis delta diagnostics, some of the details that were discussed at the delta meetings, as well as the liver meetings that we've mentioned. And we will also go through some of the current and new therapeutics in development for hepatitis delta. All right, I will stop sharing. And without further ado, we'll begin today's conversation. So to start us off with the conversation, could you please both just introduce yourselves briefly and also help us open with kind of a brief overview of the current guidelines on hepatitis delta testing and what some of the updates are when it comes to reflex testing for delta, both in the US and the EU context.
All right. Let me start with the US or AASLD guidelines, which just came out. The guidelines barely mention Delta infection in the guidelines, other than saying these are individuals who are at high risk for progressive liver disease. And they referred back to the previous AASLD guidelines from 2017 that was risk-based for testing for Delta and did not mention reflex testing. So AASLD guidelines are not applicable to this discussion, really. And the EASL guidelines really, I think, dominate, as well as WHO guidelines that dominate. So I think, Kosh, over to you. EASL is really the icebreaker on this discussion and leading the way.
Yeah, so thanks first of all for the invitation, and always happy to play the monkey to the organ grinder of Bob, because he's kind of knowledgeable and thoughtful and understands all these things. And it's great to see Super and people at this meeting. This is really important. I think starting off from the beginning, I share Bob's frustration in this role, that the society guidance always seems to be a little bit slower. And if I may say so, particularly ASLD, I think it's kind of the guideline structure per se almost sort of pauses innovation or kind of common sense. For us, I was involved in the 2023 guidance for Delta, because today is about Delta, and those recommendations were pretty clear that everyone should be screened at least once. And if there were other kind of potential concerns, there should be retesting. So I think overall, for all bloodborne viruses, and particularly this virus, which I think really is a virus that we haven't really grappled with. I think we've made huge steps over the last 5 to 7 years, but we have a lot of work to do. There can be, that repeated testing or screening and is really critical with the antibody test. I share Bob's frustration about reflex testing. I don't think we talk enough about reflex testing, and I think a major problem for Delta is that understanding of the overall prevalence because we don't have those tests coming around fast, and it's difficult to link patients in. And the example I'll give you is the sort of English-based ED opt-out program. There are other opt-out programs which in London has been finding, huge numbers of viral hepatitis, predominantly hepatitis B, but there is no reflex testing for Delta. So those patients are being lost, and Delta should be in the order of 5 to 8%. We're not quite getting there. So we would need to push for reflex testing in any lab environment where testing occurs. And number one, we should be applying testing because widespread testing should not be risk-based at all, because it is not completely a risk-based virus and issue. So I agree with Bob.
I will give you an update, though. I was giving you the bad news first, and now let's hear some good news. The AASLD has decided to have what are called expert reports on different subject areas, and the leadership has decided that an expert report on Delta is appropriate. Now, I've had some complaints about AASLD and how they do things. The Hepatitis B and Delta SIG, Special Interest Group, is supposed to be involved in all things B and Delta, but the AASLD had decided at some high level to do a Delta expert report and did not involve the SIG, which I'm the chair of. And also, I thought they should be asking the Hepatitis B Foundation to be part of that, which they did not. So I've been filing various complaints with leadership about AASLD, but there is an expert report. I did get an email last week after sending multiple complaints to AASLD asking the special interest group to propose names to be on the expert report. So I sent an email to Mark Ghani, which is a list of U.S., Canadian, and international people in the Delta space to help write that. So we'll see what their process is and how they end up selecting people. I unfortunately have never been involved with any of the guidelines for hepatitis B or Delta over the last 20 some years. They always come back and say, you have too many conflicts. Well, there's a mitigation strategy with AASLD for people with conflicts. I don't like that word. It's really disclosures. So I'm going to hopefully fight that out and lead or co-lead this process. So we'll see what works out. But the AASLD guidelines in general have always been the caboose on the train. I agree with Kosh. They have not been part of innovation. Their guidelines are not harmonized with elimination. They are just basically summaries of the highest level of medical literature and don't really format change, which is what they should be doing. So these are all things I'm working on as the head of the SIG to maybe change AASLD in general. Not more good news. Malika Toy with SAMSO at the Stanford Hepatitis Research Institute came out with a cost-effective model. Is it cost-effective to screen, including reflex testing? And that said it was definitely cost-effective to build this into algorithms. So that was really a game changer at AASLD. That's very, very happy with that. There's already data from England that Kosh mentioned, from Spain. There's US data on reflex testing that it works, it finds people. And then the final good news is uh, the four big labs in the US, LabCorp, Quest, Mayo, and ARUP, are all working on reflex testing options inside their lab ordering system. So it would become potentially a single or a double reflex. And I am going to be turning up the heat on that just after the new year. And they're all ready for our phone calls. So lots going on in that space. We will lead. We will be ahead of AASLD. We may have reflex testing before the expert report's even out. So I hope the expert report recommends reflex testing because it's already going to be there. So
Bob,
some
I'm
good
quite
news.
excited. I thought it was going to turn into a therapy session, but it sounds like you've got game here. You're doing
Oh yeah,
stuff.
we're not just complaining. We're guiding, and we're going to
Good.
make this
I
happen.
respect that. That's good on you. And I think that is a really important step in a difficult political environment and just a difficult environment completely to keep testing and to provide that access to care. And particularly I would highlight it's not just applicable to the US, but getting the diagnostic companies to fully embrace the reflex testing and the processing, and we will touch upon assays, I'm sure. Not my most favorite topic, but I can do it with you. But I think these are a really important part. All the pieces matter, just like the wire. All the pieces matter, and those are the important pieces.
Yeah, thanks so much both for sharing. So it does sound like there is some positive coming out of there, and hopefully soon we can progress to, more systematic reflex testing for hepatitis delta. I'm now going to touch on assays. HDV RNA assays were also a point of discussion, especially at the Delta Cure meeting. Could you please perhaps comment on some of the challenges that we see with HDV RNA assays and the steps maybe that are being taken to address any reliability challenges?
So shall I go first, and then you can wrap it up and be beautiful? Bob, so I find the assay discussion, and I'm going to say this because I'm a little bit unfiltered because I've just been on a plane for several hours. Um, I find the assay issue hard, in that all assays and all endpoints are going to be relevant. We've started off, and we now have two or three major assays, and a really nice piece of work led by the Hep B Forum and Heine Waldemar, just comparing those assays. And Pietro has done a lot of work. Pietro Lampertico from Milan has done a lot of work, and there are improving assays. It's the question of the availability of the assays. Robogene 2, which is now Robogene 3.0, AltoStar. Roche have been talking about an assay, but it's about getting those assays in the right environments. I think the concerning piece, and I think it does apply more widely for our interest in hepatitis B, Bob, is is that actually looking back at some samples, and I think there was a nice presentation from Pietro's group looking at three different types of those assays, the kind of credible ones, that there is a discrepancy in their operability in monitoring real-time samples. So I do think we're not yet resolved. I think the key piece, which we'll get onto later when we talk about therapies, and I think it is really good that we're now talking about more potential therapies coming into the environment clinically and potentially through the process, is whether target non-detected, what does that mean? And what's the meaning of what we're measuring? So there are two elements. Number one, are we getting a reasonable, reproducible assay that has operability? And number two, is it telling us something if it's below the target non-detected, if that's the phrase we want to use, or below a lower limit of quantification? And does that really equate to clinical meaningful endpoints down the line? So I think there's good progress in some parts, but we can always do better. And the question is what is going to be good enough? And then most importantly, what are we going to do to make this widely applicable? And that's why I love doing stuff with Bob, because it's not about one unit being really good at this. It's about a consistent, comprehensive piece where public health populations get access to the right assays after the right screening, if you see what I mean.
So we've been fortunate in the US with the development of the LDTs at the four major labs, and I was involved with all labs as they were moving that forward, including getting at least abstracts published or letters to the editor. That I think the performance characteristics are very, very high. Specificity, which really defines false positive rate, is extremely low. It's in the 1% range or less, which is great. Specificity, which more has to do with sensitivity, having to do with test sensitivity. There's a statistical sensitivity, and then where you're actually, how low you can measure virus is also very good. So if you have somebody who's viremic, you're going to find virus with the US four major assays in 96 to 99% of people. It's rare that you're going to miss somebody because of one test being positive and another test being negative at that TND threshold. So they're highly reliable where they are now. So I trust them when they are negative, and I trust them when they're positive. This is both antibody and PCR testing, which is both are very important. We know that Diasorin, which is an international organization, company with offices in the US, in Italy, and other places, should have an FDA cleared antibody test in 2026. They are in very, very late discussions with the FDA. This is public knowledge. And Roche, for quantitative RNA assay, is also in both. I think they have a CE mark now. Kosh may comment on that, but they're pending an FDA clearance that I think is highly probable after 3 years of development and discussion and arguments with the FDA on all different types of test performance characteristics. The FDA's got extremely high standards, even though they've downregulated hepatitis B to what's called a class 2, where it should be easier to get approval. And then Delta didn't really have a specific classification yet, but it really follows a class 2 designation. So we have lots of good news. We should have FDA cleared tests, having CE mark tests. And then the final step is whether companies want to go for WHO pre-qualification. I think that's in the works, but I haven't asked Christian or the woman at Diasorin on that recently.
Good.
Great. Thank you so much for those insights on assays. Another piece regarding regarding Delta testing is point of care testing. Could you please perhaps give us updates on what is the status of point of care testing for hepatitis Delta, and perhaps maybe comment on how point of care tests could change just liver health outcomes in general?
Well, I can go off and then let Bob do the detail because he's much smarter than me. I think at a space shuttle level, we need to be using to going to point of care testing. We've done that in hepatitis C. I think we could do it better in hepatitis C, for example, with thinking about elimination. If we're thinking about the big picture, there are kind of efforts in hepatitis B. Obviously, we need to have tests that are usable in all types of high to low to middle income countries. And again, we're going to have some trade-offs in hepatitis B. And likewise, we do need to think about Delta. We need to be thinking about point of care testing. There needs to be more development in that. There are efforts being made to improve it. I don't know how close we are to really getting through all of those designations that Bob has alluded to. But I think it's still a wait and see, but I'll wait to be educated more by Bob.
So there is a lot of action going on in the Delta point of care space. First and foremost is the action that has been led by Stefan Urban, who has a point of care antibody test that was developed by his team, and then the IP was sat on by his university for quite a while. But that is starting to reestablish clinical study sites. There's other point of care test companies that have published, and there were data at least mentioned or presented briefly at the Hanover meeting, then AASLD, and then again at HepDart, where I just was this last week with Pietro, Marie, Elisabetta, and Tariq, specifically in the Delta space. Um, uh, let's see. Tatiana was there also, just thinking about the Delta world, and Robert Wong. So we had a really an interesting Delta consortium. So there is point of care tests in development for both antibody and PCR. The antibody tests, the price point should be less than $7 for that point of care test. It might even get in the $2 to $3 range eventually with mass production. And then the PCR test also, including discussions about blot assays, paper blot assays, having those out and having price points of less than $20. So these are price points that are important because I work in Africa as well as other low-income countries. And if you don't get underneath those price points, it doesn't matter how good the test is, or even if it's available. If it's the price point's too high, it's not going to be used. The PCR tests, just to give you a base, if you want to get Delta PCR done in Africa, it's difficult. They do have a test kit in Benin. It's one of the best countries on the West Coast. But the PCR for Delta is over $100, which is absolutely unaffordable. It's only basically going to be used for research projects. So the good news is there's multiple kits that are being developed for rapid test point of care. And I think by 2028 or so, we'll probably have those. It's probably going to take a few more years to refine those and get them through the regulatory pathway.
Yeah, I'm gonna push back a little, Bob, because maybe it's my jet lag kicking in. I really am very supportive, and I am aware of, Stefan's kind of point of care kind of piece. I sort of as a clinical trialist, I'm still really not completely convinced by all of our correlation of the lab-based assays, which we already talked about. And I think we're putting another level of sort of not, I don't want to use the ambiguity, but sort of jeopardy, is that the wrong word? But we need to be really thoughtful about what is the line that we're crossing, and of course the price point, if we're going to get to these point of care tests. So in my head, and you're closer to this because you got to go to HepDart and all these other meetings. I think really, the way the lobbying of the companies and getting these things at the right price point is going to be important. There's going to be a trade-off with regard to sensitivity levels, and we're still not there in the lab-based kind of assays. But I think it's really important, and what you're giving us as an update is really helpful.
Yeah, there was a paper in Journal of Clinical Microbiology for antibody diagnosis of hepatitis B that was not a lateral flow. It was actually an immunochromatographic test, which is a different technology, which I think is more along the lines of an ELISA test. But there are publications. What's interesting is Florian Lemp, who was the lead author on that paper, with Stefan Urban's group, does not work in that space anymore. Unfortunately, I think he's working. I think he works with Gilead now, actually. Um,
Uh-huh.
but I think without a lead person in that space, working with Stefan, that test probably won't go forward. But there are other people who are working on this too. So I'm the eternal optimist. I think it will be out in a few years. So
Okay.
I think In Vitro is another company that's got a Delta test that's in development, just as another brand name or corporate name.
Okay.
Okay. So I just wanted to move on to kind of like hepatitis delta therapeutics now and what's been going on in that space over there. So we've been talking about functional cure in hepatitis B for quite a while now, but maybe it's from my lack of knowledge, but I haven't heard much from the functional cure aspect for Delta. Is there such a thing as functional cure for Delta currently? And with the current pipelines that's been development, are we getting any closer to achieving that?
Ooh, do you want to go first this time, Bob, or do you want me to swing at this one big time?
I think the time is right for you.
So nice softball to start off there, Super. You went really right in very hard. Ugh. So I'm going to give a completely unfiltered, being back in the back of the plane for like a bazillion hours kind of view. So enjoy the moment. Um, I I think we're at a really interesting and exciting juncture with Delta. I think to give context, remember that it's not that long ago we had no real therapies for Delta. Number one, I think it has to be said that we all agree that Delta presents late, Delta is more aggressive, Delta has an increasing risk of HCC, and at King's, which is not really relevant, but we've transplanted probably seven or eight Delta patients. So, and they all seem to be young. So we're still seeing Delta as an issue. All of the issues about the pathway of care and diagnostics kind of piece, and I think the assay piece is also relevant. So for me, um, there are two camps really, and we need to really celebrate the fact that we have a therapy that is licensed with bulevirtide, with really good kind of platform studies, the Maya studies, and then now a huge amount of real life clinical data. But our endpoints have really been a two-log drop or undetectability. See previous history about assays, and then likewise ALT normalization. So I'm sort of talking around the subject because you said, are we going to finite treat? Can we get finite treatment and cure in Delta? Well, we can in some patients, which is those who are non-cirrhotic. We can do that with pegylated interferon and bulevirtide, and I think the French data is really impressive with regard to that, number one. But by and large, because these patients present late, by and large, because I think we have a little bit of a historical, but also a sort of real life lived experience of side effects and toxicity associated with pegylated interferon. We're currently talking about longer term suppressive therapies by and large with bulevirtide. The questions I would posit back to Bob are really, are the endpoints really useful? I've always felt that the virus, if you get it negative for long enough, and we see some evidence of that with long-term follow-up of those who are on treatment with bulevirtide from Fabienne and a poster at AASLD, but the numbers are small. Or is that a really important kind of endpoint? And how long do you need to kind of be target non-detected with that mode of action of drug, an entry inhibitor? and or and or really ALT, whether ALT is really a meaningful endpoint. So when we talk about then therapeutics, I'd really like to have a discussion about the different modes of action of these other therapeutics that are coming through very fast and are really quite exciting. To say, are we really meaningfully finding something that's going to be clinically beneficial? So by and large, to answer your question, we should always be aspiring to finite therapy. I I think by curing hepatitis B or getting a clinical cure of hepatitis B, you potentially get to a clinical cure of Delta, but those drugs that we're getting into are not being seen in Delta at this moment in time due to probably safety reasons. We're currently suppressing patients because most of them present with end-stage advanced liver disease, and the pipeline is superiority to the rates that we see. But I think if we were brave enough, or if the companies were brave enough, I think we will start to see stopping trials of those larger phase three studies, and then we'll wait and see. And for bulevirtide, we will see stopping therapy in a trial type environment or real life trial environment. I wait to see because I think Delta is kind of a really difficult virus allied to those issues about assays. And I'm going to hand over to Bob there. Sorry for that kind of soliloquy.
So it is great to start with the endpoint discussion, and the two log reduction, normal ALT was what was proposed through a white paper in Journal of Hepatology. Heiner led the way. Mario Rossetto was dragged kicking and screaming to that endpoint. And it was the right thing to do. And the regulatory agencies, specifically EMEA and FDA, embraced that proposal, which was the right thing. I call it a transitional state. We were looking at data from interferon, and when you have those two endpoints, patient outcomes were markedly improved: less death, less end-stage liver disease, transplant, maybe even cancer. So that was really that was a transitional state. We are now further along. TND, target not detected on treatment, is, I think, at least the gold standard. I think platinum standard is finite therapy and being RNA negative off treatment. And I think all the companies are looking at that. You're seeing exploratory events with bulevirtide of stopping therapy and seeing if people can remain negative. But the big question is Delta curable without hepatitis B, at least functional cure? And I used to say no, 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 no for probably a decade, and now I'm saying yes, yes, yes. That this, I think the data shows that we have this large number of people who are antibody positive and PCR negative long term, right? So if you get Delta infection, whether it's acute infection, or which is co-infection or super infection, a substantial number of people clear RNA from their blood long term and do not reactivate at any time, whether spontaneously, with chemotherapy, with immune suppression, with organ transplant. This is an RNA virus. It should be curable alone without curing hepatitis B, and I do believe that happens. There's a natural cure with Delta, just like we see with hepatitis C. So in the end, I think this is going to be curable with these new therapies. And we have a number of new therapies that we can go into a little bit more detail. We have the Blue Jay antibody, which is now, last week, it was a hot topic. Blue Jay was purchased by Mirum. So Blue Jay is now inside a rare disease company. We have the HHH data that was presented also at AASLD. Also, it's a different antibody targeting a different part of the virus, I believe pre-S1. And then, of course, we go back to the Vir data that is there also super exciting, and that's dual agent. And they're showing great on-treatment viral negativity, which is, mind-boggling that we're in the 70, 80, 90, 100% range of virus undetected. So, and then looking at this, a stepwise step increase with these drugs, it's really fantastic. So if you look at just ALT and RNA negativity, ALT normalization and 2-log reduction, we're getting in close to 100% reaching that with these medications. I want to include some other technologies that are moving forward, and that's the lonafarnib and potentially even lambda interferon world that's has been reinvigorated by a company called EIT that are in this space. And a little bit further out are oral NTCP receptors. And then coming back to the interferon days is what will happen with interferon. We're seeing at least preliminary, early, exciting data with PEG plus bulevirtide, and that's our first hope at finite treatment, I think, because we're hoping bulevirtide will be approved in the U.S. Many years later than the EMEA, but hopefully by May of this year, we'll have bulevirtide available at 10 milligrams a day, which is the dose that was used for the finite therapy interferon combination studies. So huge amount going on in the Delta space. And just to wrap up with HepDart was very interesting because they were at HepDart every other year. There's a financial panel, person named Jeff Meacham, another man named Tavi from the investment community, and they're put on the hot seat for up to 90 minutes about where are they spending their money, where are they investing. And 2 years ago, we asked them about Delta, and they said Delta's not even on our radar. And yet, there's a huge amount of investment that's gone into each of these companies with the purchase of one of them by a bigger pharma recently. So Delta has really hit the big times, fortunately. And I know Gilead's led the way with bulevirtide, but we've got some great, exciting details coming out with newer technologies. All right, that's my short story.
Yeah, thank you both.
Yeah,
Go on,
can
Gosh.
I just kind of widen that? I think that is a good discussion and nicely done. Again, I want to maybe I'm the one that needs therapy, Bob. I do think it is really positive for patients and everyone in the field. Um, I think certainly the Brelovitug data and the VIR data and the other monoclonal antibody data that's come out predominantly of China and Pakistan is really interesting because they're safe, number one. But really the question is are they providing more than effect than just soaking up all of the surface antigen and then providing that? So again, I still want to kind of posit the idea of the different mode of action might impact on what we're actually wanting as an endpoint. I do think there is a huge amount of rich data from the lecanemab studies that needs to be seen by the community, and really keen to see that that's kind of resurrected. And I would ask back, yes, in this situation, Europe and the UK, we were behind Europe. We've had access to blue airtide for a long time. I do think, the French data is nice data demonstrating, the confirmation in real life of the studies to show that you can, with an immune modulator like PEG, get to a finite cure in a significant proportion of patients with Delta. So I agree with everything you're saying. I just wonder whether we are going to need to think a little bit more about the mode of action. And I would urge the companies to think wider and to think about finite rather than chronic suppression, which is kind of an easy endpoint. But it's great that that I'm making that comment in a disease that didn't have a therapy, 10 years ago.
Cool. Thank you, Kosh. Thank you both. So we touched on beloved type quite a bit there. I was just wondering, since you did mention the EU and the UK did have bulevirtide for some time now, the US is still awaiting its approval. Was there any, I guess, any additional information from AASLD and the Hepatitis D conference regarding its kind of effectiveness based on the real world data that we've seen so far?
Can I
Guess what? There was, funny enough. I mean, we put some data in, and I think again we need to congratulate Delta Cure, shepherded by the community and by Heiner and by Pietro, the sort of Save D cohort, the Shield D cohort, looking at real life outcomes of safety, effectiveness, and clinical outcomes that Bob alluded to, with a decrease in clinical outcomes, taking us now beyond the trials and taking us kind of in a real life setting. And there is a sort of wide network for Delta in Italy, particularly, showing, continued improvement, but most importantly, safety. And I think that is really important data. I would say back that in our real life experience, remember, this is a drug that is quite challenging to take as a patient. It is a drug that needs to be drawn up. It is a drug that needs to be injected subcut every day. And I think going back to those other newer therapies, they may have a additional benefit because certainly for the VIR combination, that'll be once every 4 weeks. And the Brelovitug data, which I know about, but one thing I do know about is that's either weekly or again every 4 weekly with injections. So there is I think the field is moving. I go back to your we need to see stopping data on bulevirtide, which comes back to quite long-term treatment, and then negativity persistently gives you that potential. I also do think, answering Bob's comment about hepatitis B, surface antigen positive, delta antibody positive, HDV RNA negative, we need to not forget that's a genotypes dependent piece as well. And the majority of our patients are genotype 1. A lot of African patients come with genotype 5, and they respond to interferon alone better. They have a milder disease, and they're more often negative on HDV RNA sequentially. So to come back, I think it is really an exciting time. I think it's a bit, I think, the really impressive piece is that the VIR program has taken on a phase three program from their study, which is Solstice, which was published, and they're pretty much enrolled. Certainly the two out of the three de novo studies. Um, and the Blue Jay Azure program is also recruiting very actively. So there's a lot. It'll move a lot the field, and I think there's a runway for these companies to get to where they need to get to file. That runway, Bob, might not include finite therapy at this moment in time, but I also agree the potential of an oral NCTP inhibitor, I think, again, provides a sort of injection of dynamism into this field. And we need to prepare those with lived experience and patients to be ready for these trials. And each of us have different types of patients, and really, we will only get better treatments if we're able to support the clinical trials by recruitment and quality retention. And that's a team approach with the patients being the most important.
A few more comments on bulevirtide, if that's okay, Supa. Um, so Elisabetta at HepDart showed data that may have been at ASLD or in Hanover. I wasn't sure, but it was definitely kind of hit me in the forehead that there were two things in her long-term study which stretched out more than 3 years, large number of real-world Italian patients showing breakthrough on bulevirtide at the 2 milligram dose at 15%. These were late breakthroughs. It was unclear because no one's found any resistance, and there's a theoreticals that this is some change in the receptor activity, and that was, I thought, fascinating. Um, Also, TND in that real world study, this again long term getting out in the 3 year range, was 30%. That's the best that I have seen so far. Kosh, you may know some other numbers. So that was one of the most interesting things at HepDart, and I'm so happy Delta was such a hot topic there. Back to what's going to happen when bulevirtide gets released in the U.S. The reason there was this long delay is that the FDA basically told in no uncertain terms that their preparation steps that Kosh talked about was too much for American patients. We're not as smart as European patients. We need something very simple. So they've come up with, I believe, in the end, this is something I'm just reading the tea leaves. I don't know inside information from Gilead, but I'm sure they're going to have a device that's similar to the Ozempic, the GLP, where it's, you push a button, it mixes, you push a second button, and it injects, something like that. They'll be super easy for patients. And the same nice thing is they're going to go from 2 to 10 milligrams, and it's again one volume. So they had to figure out how to get 10 milligrams into the same volume as 2 milligrams, so it would be attractive to patients to inject without excess amount of pain. Finally, safety, safety, safety. The only thing that you see with the safety signal with bulevirtide is elevated bile acids, but this has not been clinically relevant. Fortunately, there's been no liver tox signal with any of these medications, which is good news. But we've got a lot of good news out there because, long-term injection, the investors love chronic therapy. That's where they make the most money. The patients want finite therapy, and that's where there's going to be the greatest uptake when you offer finite therapy. So we've got this yin and this yang going on between the investors and the patients. But I think in the end, if we're good providers, the patients will win, and we'll also convince the
The elite
investors
British
to
private school that
invest.
lost its way in Singapore. My Madison marriage and
Oh,
Antonio
sorry about that. Whoa.
Singapore.
No worries. No, thank you both. One last question on, I guess, bulevirtide, and then we can move on to the next one. So, Kosh, you mentioned a lot about the stopping bulevirtide therapy. What considerations, what do we need to look out for? What does that look like if we were to do that? Is it similar to, like, say, hepatitis B, we're stopping nukes, or is it going to be much
So
more
a lot
to?
of I think we need to see proper data, and we're going to see data on a controlled bulevirtide stop study, and I'm on the safety board of that. So I do. I do think it's a watch this space piece. I also think we can't talk about predictors. There's been some suggestion in the early days that there can be potentially a flare stopping, and that could be potentially detrimental for those patients with cirrhosis. Let's wait and see. Um, do I personally think that bulevirtide is a good enough agent to maybe give us a finite therapy piece if, without with only a publicly available data, I'm not sure. For us in our unit, we really, have jumped at using bulevirtide, but for us, it's for those patients who cannot get into clinical trials or for other reasons. So again, what I would reflect is different populations of patients in different areas have different characteristics, and I've always said this is that the long-term Delta patients, stereotypically from Italy, Milan, who've been under the care of Pietro and other really good doctors for a long period of time. That is a different type of patient to what I see. We see younger patients, we see patients related to migration, but other ways of getting acquisition of Delta. They may be more mobile, they may have more requirements of understanding their condition. So I think we need to not leave anyone behind when we talk about therapies. And I think getting a wider access and certainly getting FDA approval is pretty important, I'd say, for the US, because I think once you have a treatment that drives diagnosis, education, screening, the whole piece, which I think is really good for patients, by the way, and those with a lived experience. But I think for me, I don't know whether bulevirtide as a first generation NCTB inhibitor is going to be good enough in those with advanced disease for us to be able to do that. But let's travel hopefully.
I have a suggestion, Supa and Stella, as the moderators. We moved very, very quickly into therapies, testing and therapies, but we moved past epidemiology. I know we have maybe 11 minutes left, but can we talk about epi just for a moment?
Yes, of course, let's do it.
So there's constant discussions about how much delta there is in the world, how much delta there is in different regions, and this is a hot topic in the US because multiple companies have been turned down for having orphan drug designation because the FDA Orphan Drug Office said, oh, this is no longer an orphan designation because the epidemiology, the prevalence of infection is far over 200,000. And they based that on two manuscripts, one of which is from Patel and David Thomas's group, and Hopkins in collaboration with N. Haynes. But the assay that was used for that specific epi study, specifically antibody test, has actually been withdrawn from the market because of specificity problems. N. Haynes agreed to retest all those samples with a state-of-the-art assay. And I think the assay they used was the CDC assay because the CDC came up with their own antibody and PCR test. That data is pending. We still, because of the instability of our current leadership in our government, has really destabilized the CDC. But I'm hoping in the next few months we'll have a reassessment of that Patel paper and the N. Haynes to try to fix that problem. Because we, as KOLs, believe the prevalence of Delta in the US is less than 70 to 80,000. There's numbers as low in the 25,000 range if you look at the recent Quest data set that Elizabeth Marlowe was lead author on. So we think there's been overestimates. You probably remember there's that Taiwan Review of the World Literature paper where they were saying there were 60 or 70,000, 60 to 70 million people with Delta in the world. So we really think it's a lower number, and probably because so many people have died of this disease. I think that's one impact, as well as inaccurate assays. So there's lots going on in epi. Also in the public domain, this was discussed at HepDart with the investor panel, And when people said, well, what did Miram pay for Blue Jay? And that was, I'm just going to give you some general numbers in the $300 million range, which is a relatively low number compared to what Gilead paid for bulevirtide, which was over $1 billion. But then they said, well, why did they pay this number and what's going to happen next? And apparently, when they did the modeling at Miram and deciding what the real market size was in the US, they said, no, there's only 15,000 people in the US with Delta. That's way too low, in my opinion. But they were conservative with their cost modeling, which is smart, of course. So, what's the real number? We're not sure. We've got numbers ranging in the 20,000 to 80,000 range, but it's a universally fatal disease. So, you can have a diagnosis, you're going to die from Delta. So, every one of those person becomes a candidate for either chronic or finite therapy eventually. So, epi is important. Alex Stockdale from Liverpool, I think, right, Kosh? That's where Alexander's based. Has been the lead person in the world on epi, and he had a fantastic presentation back to Hanover, the Delta Cure meeting, on what Delta looks like today. And he's bringing in more information, especially from Africa and Asia, Central Asia, on epi data. And we're less than 15 million globally. That, I believe that's the fact. So, quick summary on that.
And no, I always will try and agree with you, Bob, and I think it's really important when you've not had access to the therapies and in North America. I still think there are some real anomalies when you talk to our colleagues from Pakistan, and Mongolia, and you've traveled and been there. I don't think we really fully understand the epi and the prevalence, and how in some villages it can be very high and in others it can be low. And I just don't know. I have to be honest, I don't know. I mean, certainly in London, historically, a long time ago when I arrived at as a consultant, one of my colleagues, Phil Harrison, did some work, and it was 8% of antibody positivity. And we are not in a position to give you more information because we haven't done it for London again. So I think we do still need to understand what we've got. And hopefully, the more focus there are on therapies or the availability of therapies, the better our diagnostics will be to be able to empower those sorts of pieces. And I feel, Africa is a hole in where we really understand the data. So it's good to talk about North America, but globally, I think we still need a bit more sense of what's going on, and that's going to take a lot of work, I'd say.
There was a chat comment from Andre Reinhardt from RoboGene in our conversation here about their willingness to work in Africa. And Africa is interested in Delta. We definitely need to do some pilot projects. And now that we have West Africa wired with our Smile Coast of Africa project, I'm working on with the people at Hepatitis B Foundation, Catherine Freeland, Brenda Evan, who works for me directly, but is collaborating also, Noah Trudeau. It's really, we're going to make a difference in West Africa and hopefully a model for more of Africa over the next few years. It was interesting, in Africa, they have very little data on Delta because they have very little data on hepatitis B. The lack of testing and, of course, lack of treatment is sort of the bookend to that. But we will make progress in West Africa and across Africa with these collaborations. We're getting everybody to talk to each other. It's not just 20 countries here or 15 countries there. We want to get the 54 countries in Africa all knowing what everybody else is doing. COLDA, SOLDA, there's just a whole number of organizations that are going to help Africa move forward and let Africa lead Africa with our help.
Yep, thank you so much both for sharing those insights. And we look forward to some of the data from West Africa. Dr. Gish, I was wondering also if you can use up 2 minutes perhaps to also update us on any new data from Mongolia, because we know that it's one of the regions with the highest burden of HDV, and there could be some data updates from there.
Right. So I spent 5 days there just before the Delta Cure meeting because I travel around the world. It was interesting flying over Russia on a Mongolian airline as part of that Circle the Globe tour. But I'm going to say that the numbers that you've heard in Mongolia in terms of prevalence of B and prevalence of Delta in the B infected population. So B 10 to 20%, Delta in B is about 70%. That number hasn't changed, but we know the Delta is the leading cause of liver cancer, Delta plus B, in Mongolia, which is the leading cause of cancer in Mongolia, which is a leading cause of death in Mongolia, and it has the Mongolian government's attention. I met with the Minister of Health when I was there, met with the NCCD, their CDC equivalent and research unit, and we're getting pharma interested in working in Mongolia now that Mongolia legally has a path forward for drug development, specifically phase 3. So many pieces are falling in place in Mongolia, which is great. Just to remind everybody, Mongolia is a democracy caught between two large countries that are, they're in a challenging situation. But now there's a United airline flight every day to Mongolia from the West Coast. So there's going to be an infusion of interest. The China team from HHH has actually worked with Mongolia to help recruit patients for their HHH antibody trial. So Mongolia is doing well. Meredith Potts, head of fire, is there. The Onum group has done work, especially working with our team from Sweden. So there's a lot of activity. Mongolia is moving ahead and should help be a model for the rest of the world.
Great, thank you so much. Um, I do not see any new questions from anyone in the audience at the moment. So with that said, I would like to say thank you all for joining us today, and thank you to our excellent speakers for joining this webinar very early for Dr. Gersh and very, very late for Dr. Agarwal. We truly appreciate that. And a recording of this webinar is going to be available on YouTube for anyone to revisit. And yeah, we thank you so much. And we encourage anyone who's interested in hepatitis Delta to see our Hepatitis Delta Connect project here at the Hepatitis B Foundation. And with that, thank you so much.
Thank you very much. Always good to do good stuff.